Histology is the study of tissues. A tissue is a group of cells and stuff from the cells, usually coming from the same part of the embryo and working together to do some sort of role in an organ. On our grand hierarchy of things, we have atoms and molecules and cells forming tissues. So cells come together to form tissues, tissues come together to form organs. There are four primary tissue classes: epithelial, connective, neural, and muscular tissue. Everything is a subset of those four classes. Now, you're not just cells, you're cells and the stuff from the cells. Cellular entourage, the crap the cells bring with them, cellular luggage. It's called the cellular matrix or the extracellular matrix. We've got water, usually some fibers, some proteins. There's this sort of gel fluidy stuff we call ground substance or interstitial fluid if it's in between the cells. This is all extracellular material or outside the cells. Depending on the tissue, it may be fluid. If we're talking about blood, it could be fibrous if we're talking about connective tissues. It could be ossified if we're talking about bone. It varies from tissue to tissue. Epithelial tissue is just cells. Now, epithelial tissue is one of our four primary tissue classes. And like I said, it's just a bunch of cells. We don't see an extracellular matrix here. They are sheets of cells. The outer surface will be exposed either to the external environment or to an open space in the body. Think your skin or the lining of your digestive tract. So it covers your body surface, lines all the cavities, and forms the lining of most hollow organs. Lots of glandular tissue is also epithelial tissue. Now, there are 5 characteristics of all epithelial tissue, and we'll talk about those now. The first is polarity. Polarity means that there are 2 sides to every epithelial tissue. There is a basal surface. The basal surface sits on top of what's called the basement membrane. And the basement membrane holds the epithelial tissue to the connective tissue beneath. The surface facing away from the basement membrane is called the apical surface. So here's our basic epithelial cells here. So here is our apical surface. This would be an open space in the body. And then here is our basal surface down here, lateral surface of the cell in between the cells on either side, basal surface down here. Epithelial cells are also held together by specialized contacts. They form continuous sheets. They're held together with tight junctions. And if you'll remember from when we talked about cells, tight junctions tightly hold the cells together so that stuff doesn't leak in between them. And desmosomes. Desmosomes physically hold the cells together in continuous sheets so that if tension is applied, it doesn't pull the cells apart. So there we see our cellular junctions between those 2 cells. Epithelial tissue is always supported by connective tissue. There is a basement membrane which sort of glues the epithelium to the connective tissue underneath. It's filled with collagen and some glycoproteins to hold things together. Epithelial tissue is avascular, but it is innervated. Avascular means there are no blood vessels going through the epithelial tissue. But it does have nerve fibers that supply it. Think about the epidermis, the outer layer of your skin. It is avascular. You've had this, you've had the paper cut that doesn't bleed, but it still hurts like hell because it's still innervated by nerve fibers. You can still feel it, but it doesn't have blood vessels running through it. So epithelial tissue, avascular, but innervated. Finally, epithelial tissue regenerates. It will grow back. Epithelial cells because they are in contact with the external environment or to an open space in the body, will continually die and continually be replaced. There are two major types of epithelial tissue: simple epithelial tissue. Simple epithelium means there's one layer of cells. Every cell touches the basement membrane. Stratified epithelium is layers of cells where cells are sitting on top of cells. Not all the cells will be touching the basement membrane. Some of them will be on top of other cells. So here we see simple epithelial tissues and the different shapes of the cells. Squamous means flat, cuboidal means cuboidal, and columnar is column shaped. Underneath, you see stratified epithelium, and you can see those multiple layers of cells. Now, simple epithelium is one layer of cells, and there are three main types of simple epithelium: simple squamous, which thin, flat cells; simple cuboidal, which are square or roughly cuboidal, or columnar cells. There is another type called pseudostratified. Pseudostratified means fake stratified. It looks like it's stratified, and you can see that way down here. It looks like it's stratified, but it's not. The taller cells are overlapping the shorter cells. They are all actually still in contact with that basement membrane. Simple squamous epithelium is found where you need something to cross that barrier very quickly. It is a single layer of cells that are very thin and flat. So where we need rapid diffusion or filtration, we find simple squamous epithelium. This presents minimal barrier. So we find this as the endothelium, which is the lining of blood vessels, the mesothelium, which lines the body cavity, covers organs. We find this in the alveoli of the lungs, so that oxygen and carbon dioxide can cross through that barrier very quickly. We don't want multiple layers of cells here because it would take longer for those things to cross through that barrier. So here we see simple squamous epithelium. Thin, flat cells in a single layer. Simple columnar epithelium is also associated with absorption and secretion because we just have one layer of cells. Often we see microvilli or even cilia here. Cilia to move something like mucus along the surface of the cells, microvilli to increase the surface area of the cells. Whenever you see microvilli, you need to think absorption because microvilli increase the surface area. And the only reason to do that is if you're moving some sort of product across that membrane. Microvilli, absorption. Here is the simple columnar epithelium in the lining of the small intestine. And you can see those microvilli there. Pseudostratified columnar epithelium, still absorption and secretion. Here, like I said, the taller cells overlap the shorter cells, so it looks stratified. Here we see pseudostratified columnar epithelium in the respiratory tract. We see cilia there, which would move mucus along the surface of those cells. Goblet cells are often associated with simple columnar and pseudostratified columnar epithelium. Goblet cells make mucus and secrete it onto the surface of those cells. Simple cuboidal epithelium we find in the walls of the tubules of the kidney, the smaller glands. They look like this. Here's cuboidal epithelium in the uh, distal convoluted tubule. Still, because we have a single layer of cells, the goal is to move the product across these cells very quickly. Now, stratified epithelium, we have a different goal. Between 2 and 20 layers of cells, still we get all three shapes and something else called transitional epithelium. But for the most part, stratified epithelium is not there for absorption secretion. Stratified epithelium is there for protection. We have multiple layers of cells to protect something. Stratified squamous epithelium is the most abundant epithelial tissue in your body. And there are two types of stratified squamous epithelium. Keratinized. Keratinized, we have a layer of dead cells on the surface that are full of keratin. Keratinized stratified squamous epithelium is your epidermis, the surface of your skin. Then we have non-keratinized stratified squamous epithelium. These cells will always be kept moist for friction reduction. They are there to resist abrasion. Think the lining of your mouth. Those are stratified squamous epithelial cells. So like when you get the paternity test in the mail from your lawyer, you have to scrape the inside of your mouth and then send off that little test tube. Not the father, yay. Actually, yeah, I got 5 kids. It's always me. Those are stratified squamous epithelial cells, non-keratinized. So here we see that in the lining of the mouth. Here we see keratinized stratified squamous epithelium on the surface of your skin. There is stratified columnar and cuboidal epithelium, but they're not very common. And you're not going to find more than 2 or 3 layers here. We get like 20 layers on the stratified squamous epithelium. Here, you might get to 3 tops. Stratified cuboidal epithelium is really rare. It would only be in the ducts of some of the bigger glands. And stratified columnar epithelium would also be pretty limited. Found then again in some of the glandular ducts. So here we see stratified cuboidal epithelium, and you'll notice it's 2, 3-ish layers, and this is in the duct of a sweat gland. And here is stratified columnar epithelium in the duct of a salivary gland. Transitional epithelium is typically associated with the lining of the bladder. It's called transitional because it changes shape. It varies in shape based on how full your bladder is. So when the bladder is empty, the cells are kind of fat and cuboidal. And when the bladder is full, it stretches them out and they look more columnar. So here you see this in the empty bladder. And they're really kind of fat and cuboidal. And then as the bladder fills, it will stretch these cells out and they look a lot more columnar. All right, so let's practice. What kind of epithelium is this? If you answered simple cuboidal epithelium, well done. Way to pay attention or look ahead on the slideshow. What's this one? Simple columnar epithelium. Again, well done. You like how I assume you got this right? You're welcome. What are these cells? I ask as if you can answer. Goblet cells. Goblet cells make mucus. This is actually a fairly poor example of simple squamous epithelium. This one. This one is pseudostratified columnar epithelium. Now, when you look at this, you're looking at these and you're like, well, how can I tell that this cell, for instance, is still in contact with this basement membrane? Why is this not stratified? And my answer there, 2 things. One, the presence of these goblet cells. Goblet cells will always be with some sort of simple epithelium. If you see that goblet cell and you can't tell if it's stratified or pseudostratified, it is pseudostratified because you won't see goblet cells with stratified epithelium. The other thing here are the cilia. Again, you won't see those with stratified epithelium, just with pseudostratified or simple epithelium. Remember that pseudostratified is fake stratified. It's still simple epithelium. This one, if you can see the little inset down here, it comes from your foot. So this is stratified squamous epithelium. We see all those layers of cells, and all these cells out here are dead. So that's keratinized. This one. Again, if you see the subset down here, this is from the lining of the female reproductive tract, and you see all these layers of cells, which makes that stratified squamous non-keratinized. All right, this one, more than one layer, stratified. What shape? Cuboidal, in the duct of our sweat gland. This one is tricky. In the umbilical cord, these cells are fat, but as that umbilical cord stretches and pulls, they may look more columnar. This is transitional epithelium. Now, epithelial tissue is also found in glands. A gland is a group of cells that are making, and sometimes it's just one cell that's making and secreting some sort of product. We have two types of gland, endocrine and exocrine. We're not going to spend any time with endocrine glands because that's for another semester. Right now, we'll talk about exocrine glands. Exocrine means that we're secreting the product either onto the skin or into an open cavity of the body. So, mucus, bile, sweat, oil, saliva, digestive enzymes, those are all exocrine products. And there are two types of exocrine glands, unicellular and multicellular. A unicellular exocrine gland would be a goblet cell. It's one cell making mucus. Multicellular exocrine glands get classified by their structure and their mode of secretion. Most multicellular exocrine glands are called merocrine glands. A merocrine gland releases its product through exocytosis, meaning it makes the product in the cell, puts it in a vesicle, and pushes it out of the cell. Now that we do have holocrine glands, a holocrine gland will produce a product but not secrete it. Instead, the product just accumulates inside the cell until the cell explodes, and now it's secreted. Apocrine glands are iffy. We do know that they exist in certain animals, but their presence in us is uncertain. In an apocrine gland, just the tip of the cell will break off. We can also classify these glands by their structure. We have simple tubular, like glands of the intestines, simple branched tubular, and the gastric glands. These are simple because the duct, as it comes out, is not branching. You've got one duct for all three of these little branches down here. Down here we have alveolar, they're sort of berry-shaped here. Simple alveolar, there really aren't any in people. And then here are like sebaceous glands. This is a simple branched alveolar. A compound duct, we have 1, 2, 3 ducts that come into one common duct here. So this is a compound tubular duct found in the duodenum. Here's a compound alveolar duct, like in a mammary gland, and a compound tubular alveolar duct, which is some of both, like in a salivary gland. Here we see merocrine glands secreting their product through exocytosis. Hypothetically, an apocrine gland where just a part of the cell breaks off, and a holocrine gland where the whole cell just explodes. Now, those are all epithelial tissues, just a bunch of cells. Connective tissues are different. Connective tissues, the cells are spread much further apart, and mostly it's made up of extracellular products, fibers, and ground substance. Connective tissues are the most abundant, the most widely distributed, and the most variable tissues. There are 4 types of connective tissue. Now, before we do that, let's go back and review. There are 4 types of tissues, 4 primary tissue classes: epithelial, connective, neural, muscular. Now, there are 4 subtypes of connective tissue: bone, cartilage, blood, and what we call connective tissue proper. Connective tissue holds things together. It holds you up. It's protection. It's your immune system. It's movement in terms of leverage, not muscles by any means. Storage of stuff, body temperature, transport of oxygen and carbon dioxide. Right now, we're going to focus on connective tissue proper. The cells of connective tissue proper are called fibroblasts. Fibro, as in fibrous, and anytime you see that suffix blast, that's a cell that's laying down extracellular material. Also associated with connective tissue proper, we'll probably see macrophages, leukocytes, plasma cells, mast cells. All of those things have immune function. Leukocytes are white blood cells. Macrophages are cells that will eat other cells. Um, Plasma cells make antibodies. Mast cells release histamine. Adipocytes are fat cells. But all of those cell types are associated with connective tissue proper. There's lots of extracellular material. The fibers will be collagen. We have reticular fibers, which kind of make a mesh, and we have elastic fibers, which can stretch and rebound. The ground substance, the sort of gel that's surrounding all of this, will be made up of a protein carbohydrate blend, these things called glycosaminoglycans or proteoglycans, and some adhesive glycoproteins, much like we'd find in the basement membrane. Now, there are two major types of connective tissue proper: loose connective tissue. In loose connective tissue, there's a lot of ground substance. Cells, some fibers, lots of ground substance. And then there is dense connective tissue. In dense connective tissue, it's mostly fibers. In fact, the fibers take up more space than even the cells. Here is areolar tissue. Areolar tissue, the fibers are not very organized. There's lots of room for blood vessels to go through here and lots of empty space. Connective tissue, unlike epithelial tissue, is typically vascular, meaning there are blood vessels going through it. Now, there are exceptions. Obviously, blood does not have blood vessels going through it. It is blood. And cartilage is also avascular. But because we've got space here, we've got lots of space for these blood vessels. Reticular tissue is a mesh. Here we see a cross section of the spleen and lots of these white blood cells, leukocytes, on these reticular fibers. Adipose tissue is fat tissue. The major cells are adipocytes. And again, there are lots of blood vessels going through here. So loose connective tissue, lots of room for blood vessels. Dense connective tissue is mostly fibers. Now, while it's not an avascular tissue, when we look at this tissue, and this is dense regular connective tissue, the fibers are running in parallel, there's not a lot of room for blood vessels. So while it's not avascular, it doesn't have a giant blood supply. Most of the room is taken up by fibers. The cells will be fibroblasts, making that extracellular material. This is what tendons and ligaments are made out of. Dense, irregular connective tissue, still lots of fibers, most of the space taken up by the fibers, and they're sort of randomly oriented. We would find this between the skin and the muscle underneath. So here we're looking at a section of the dermis, I believe. Cartilage is avascular. We'll talk a lot more about cartilage later, but there's no blood vessels in cartilage. We have sort of a gel matrix, and the major cells here are chondrocytes. Again, more on cartilage later. Bones are calcified. Here, our extracellular matrix is bone. More on this later. Blood is still connective tissue. Our sort of definition here for connective tissue is cells with a lot of extracellular material, ground substance or fibers around it. So here we have blood. Our cells are called formed elements, and the ground substance is a liquid called plasma. Neural tissue and muscular tissue are both excitable tissues. Nervous tissue is made up of neurons and glial cells. Muscular tissue made up of muscle fibers. More on both of those later. Tissue will be repaired either through regeneration, meaning it grows back like epithelial tissue, or through fibrosis, which is the formation of scar tissue. The skin will use both, as we have epithelial tissue on the surface of the skin and this dense, irregular tissue beneath that. So here you see the healing of the skin where we have regeneration out here and fibrosis down here, the formation of that scar tissue. Now, that will rebuild over time. Scars fade. While it may never come back to exactly the way it was, it will fade over time. Histopathology is the study of tissue to examine diseases. We would take a tissue sample from a biopsy or an autopsy or surgery and then examine those tissues. If you're interested in the medical field, but you're not interested in, dealing with people, histopathology may be your gig, because here you still get to make these diagnoses. You're looking at cells, you're diagnosing cancer, this sort of thing, but you don't actually have to see people, like you're back in the lab. Those shows like where the doctor like discovers that you have cancer. That's really not the case. Like your doctor sends that sample off to somebody in the lab who looks at it under a microscope and then sends him back a piece of paper that says you have cancer. To study tissues, you have to take them out. Then you have to fix it or preserve it. And then we stain it. Obviously, these slides of tissues we've been looking at, your tissues aren't pink or blue or purple. They're all sort of beige-ish, sometimes clear. So we stain it so we can actually see the cells. So some terms we'll use throughout the semester. The first is atrophy. Atrophy is the shrinkage of tissue, either because we're losing cells or because the cells themselves are shrinking. We have atrophy because of malnutrition and the tissues are wasting away. We have what's called disuse atrophy, especially in the muscles, where the nerves no longer supply the muscle. So without any use, the muscles will atrophy. The rule of atrophy is if you don't use it, you lose it. And any tissue can atrophy. The brain will atrophy. Necrosis is the premature death of tissue. Necrosis is caused by many things, most often infection or lack of circulation, sometimes toxins. But more often than not, it will be an infection or a lack of circulation or a combination of the two. Any tissue can also become necrotic. Here we see necrosis of all the tissues in the leg. Skin, muscle, bone, it's all necrotic. This degree of tissue death was caused by a lack of circulation. If I remember correctly, this person was bitten by a snake called a fer de lance, very venomous snake. And to save him, they tied a tourniquet around his leg, which kept the venom from circulating into his bloodstream and killing him, but it also cut off circulation to the leg and killed it. These other two pictures of necrosis are courtesy rattlesnakebite.org, which is a thing. First of all, one of the things I'll point out to you here is that they are both on the hand. No judgment. I'm just saying they're both on the hand. In my experience in post-surgery clinic, every rattlesnake bite that I saw post-surgical was on the hand. So we see necrosis here, and you see necrosis beginning here in the middle of the palm. Now this is not necrosis out here. That is surgically intentionally done. That is called a fasciotomy. The suffix otomy means surgical incision into, and fascia refers to the fascia, the tissue between the skin and the muscle. As that rattlesnake bite venom destroys cells, your immune system responds with inflammation, especially in the extremities. That inflammation can cause circulation to be cut off as the tissues swell and start to push on blood vessels, compartmentalizing the limb. So to prevent that, the surgeon here has done a fasciotomy and allowed the deeper tissues to swell outside the bounds of the skin so that circulation continues and we don't get this effect where tissues die because of lack of circulation. Gangrene is another form of necrosis. We see diabetic gangrene of the foot. The term gangrene is often used when it's caused by a bacterial infection or when it encompasses a considerable amount of tissue. Gas gangrene of the mouth there due to bacterial infection. An infarction is the sudden death of tissue caused by a sudden loss of oxygen supply. Most often when you hear the term infarction, you hear it in terms of a myocardial infarction, which is what we see in this upper left picture, cross section of the heart. So this myocardial infarction, we have the necrotic tissue here, and then we have fibrotic tissue in white over here. This left very little of the left ventricle functional, which would have obviously killed this person. You knew that because we're looking at a cross section of his heart. So hopefully didn't spoil anything. Down here we have a cerebral infarction, apparently due to some sort of head injury. And this actually was a testicular infarction, which are typically asymptomatic, but noticed in dissection. And we see that fibrotic tissue here along the testis. This is necrotic tissue after a myocardial infarction. We see little bits of cells out here in between cells. We don't get the definition that we normally would see in muscle tissue. Now, cancer is something else that we would look at in histopathology, and that happens when cells are dividing uncontrollably. Cancer tumors are classified as benign or malignant. Malignant cancers are the ones that we're concerned with because those cells tend to move or metastasize to other regions of the body. Often what comes up is what causes cancer? Someone will ask me. Obviously, if I knew that, I'd be relaxing in my beach house somewhere instead of lecturing on YouTube. The thing with cancer is it's you. They're your cells. They're just not doing what they're supposed to. So a lot of cancer is just genetic. There are genes that we call proto-oncogenes. Onco is cancer or tumor. Proto is early. And these may never come into play. But if they're damaged by a carcinogen is something that tends to cause cancer, it will convert these proto-oncogenes into oncogenes. And then we have this uncontrolled cellular replication. Luckily, we have other genes built in to help us, tumor suppressor genes. And those genes are activated should that proto-oncogene be damaged. And it causes the cell to die. And then you don't have cancer because the cancer cell is dead. About half of cancer involves malfunction of 2 of the 15 tumor suppressor genes. This is a graphic from a hypothesis that I was reading not too long ago. And it involves what's called the p53 tumor suppressor gene. In a normal cell, if the DNA is damaged, that p53 is activated and it causes the cell to die, no cancer. In an abnormal cell where that p53 gene is abnormal, if the DNA of the cell is damaged, there's nothing to prevent it now. It keeps going. This article was about using a viral vector to infect cancer cells. Around half of us will develop some form of cancer in our lifetime, and about 20% of us die of cancer. Treatments for cancer, of course, involve removal of the tumor, radiation, and chemotherapy to kill those abnormal cells. Treatments are unique and they're tailored for the individual because everybody's cancer is individual for them. They're your cells, just not functioning correctly. Apoptosis is this programmed cellular death. Whereas necrosis is the death of tissue, apoptosis is when cells die because they're supposed to. For instance, those cancer cells dying, tissues that are no longer functional. We'll see apoptosis of the cells of, for instance, the webbing between your fingers in utero. So, unlike necrosis, apoptosis is generally considered to be a beneficial thing. So, that will conclude this for histology. A little later, we'll have a review for your first exam, as this does end the material for exam one. So, look for that review. And if you have questions, just let me know.